Ever wished you could like pinpoint a specific building in a bustling city at night? Well, that's kind of what fluorescent probes do, but for cells. It's like they act as these tiny beacons, illuminating the otherwise invisible world within.
Yeah, it's remarkable how much these techniques have really transformed our understanding of cell biology. We've gone from these like static snapshots to really dynamic, real-time insights into how cells function, how they interact, and even perish.
Okay, so let's break it down a bit. What exactly are fluorescent probes and what makes them so special?
Well, at their core, they're these molecules with a unique ability. They can absorb light at a specific wavelength and then emit it at a longer one, creating this glow when you excite them with the right kind of light. And what's really fascinating is that we can design these probes to target very specific components within a cell.
So it's not just about illuminating the entire cell. It's about highlighting specific structures or molecules of interest. That's pretty cool. So how do we actually capture these glowing signals then?
Specialized microscopes are key here. They're designed to excite the probes with the right wavelength of light. And then they detect that emitted fluorescence, which creates these just stunning visuals where the targeted structures like pop against a dark background.
Okay, I'm starting to get it. But why is this all so important? Where do these fluorescent probes really make a difference in cell biology research?
Well, their applications are incredibly diverse. One major area is in identifying different cell types based on their surface markers. So think of it like attaching glowing name tags to individual cells based on what proteins they express on their surface.
So we can differentiate between the different cell types just based on their unique fluorescent signature. This must be super valuable in immunology, for example.
Absolutely. In immunology research, fluorescent antibodies are used all the time to label specific proteins on immune cells. And this allows us to, tell the difference between, say, helper T cells and cytotoxic T cells. Both of those are crucial players in our immune response.
Wow. And we're not limited to just the cell surface, are we? Can we also visualize structures inside the cell?
Oh, for sure. We have a whole arsenal of fluorescent dyes that can actually target and highlight different organelles within a cell. Like, say you want to examine the intricate network of the endoplasmic reticulum, or pinpoint the mitochondria, those energy-producing powerhouses. There's actually a specific dye for each of these structures. We can even track dynamic cellular processes like apoptosis, which is programmed cell death.
So it's like having a toolbox filled with different colored highlighters, each one designed to illuminate a very specific part of this like cellular landscape. And this ability to track these dynamic events, like cell death, this has got to be crucial for understanding both normal development and also disease processes.
Exactly. Apoptosis plays this critical role in shaping tissues during development and also getting rid of those damaged or unwanted cells. And fluorescent probes let us pinpoint different stages of this process, which gives us valuable insights into how it's regulated and also the consequences when it goes wrong.
This is already mind-blowing, and we've only just begun to explore the possibilities. But before we go further, I'd love to understand the different types of fluorescent dyes out there. What are some of the most commonly used ones, and what makes each unique?
So one of the most iconic dyes is DAPI. It emits this bright blue fluorescence and it selectively binds to DNA. So it's a staple for visualizing cell nuclei. It paints this vivid picture of the genetic material inside cells.
Yeah.
And this ability to actually visualize the DNA directly, it's been a game changer for fields like genetics and cancer research.
It's amazing how just a single dye can kind of unlock so many secrets, all hidden within a cell's nucleus. So DPI paints the picture in blue, but what about other colors? Can we use a whole palette of fluorescent dyes to reveal even more about the cellular landscape?
Oh, absolutely. We've got a wide array of dyes at our disposal, each one with its own unique properties and applications. For example, the rhodamine family of dyes, these emit this vibrant red fluorescence, and they're often used to track those dynamic cellular processes, or you can even use them to pinpoint specific organelles, like mitochondria.
So we can use rhodamine to actually see those tiny cellular powerhouses in action, capturing their movements and interactions right there within the cell.
Exactly.
That's so cool.
Yeah.
What about green fluorescent dyes? Are there any that kind of stand out as being particularly useful?
Oh, well, one of the real workhorses of fluorescence microscopy is FITC, fluorescein isothiocyanate. It's this bright green dye that's often conjugated to antibodies. This allows us to target and visualize very specific proteins of interest.
So by combining FITC with antibodies that actually recognize, let's say, a particular protein, we can essentially create like a custom-made probe, right? And that would only highlight that protein within the cell.
That's a great way to put it.
That's incredible. It's like having a molecular spotlight. You can shine it only on the actors you want to see on the cellular stage.
Yeah, and speaking of a wide range of colors, we can't forget about the Fluor dyes. They come in this dazzling array of colors, each with a distinct emission wavelength, and they're known for just being exceptionally bright and stable.
So Alexa dyes offer this kind of enhanced brightness and stability compared to other dyes. What kind of advantages does that give researchers then?
Well, their brightness allows us to see even faint signals, and then their stability means they can withstand these prolonged imaging sessions without quickly fading away.
Right.
So that makes them really valuable for experiments that need high sensitivity or long-term observation.
So it sounds like Alexa dyes are kind of like the premium high-performance option for fluorescent imaging. With so many different dyes and applications to choose from, how do researchers decide which ones are best for their particular experiments then?
That's an important consideration. Selecting the right probes depends on several factors, like the specific target they want to visualize, the type of cells they're working with, the resolution they need, and even practical stuff like budget constraints.
So it's kind of like a chef choosing the right ingredients for a specific dish?
Yeah.
The right combination of probes and techniques, that can make all the difference in the success of an experiment.
Exactly. And just like a chef might experiment with different flavor combinations, cell biologists often try various dyes and techniques to really optimize their imaging experiments.
So speaking of techniques, we've talked about microscopy a bit, but are there different types of microscopes used for fluorescent imaging? I imagine not all microscopes are created equal.
You're right. The most basic type is widefield fluorescence microscopy, and that just illuminates the entire sample at once. It's great for getting an overview, but it might not be ideal for visualizing fine details or structures that are really deep within tissue. For higher resolution images and the ability to actually optically section through these thicker samples, we've got confocal microscopy.
So what are the key differences then between confocal and widefield microscopy? And why would a researcher choose one over the other?
Well, confocal microscopy, it uses a pinhole to get rid of that out-of-focus light. And that results in much sharper images and even the ability for us to create 3D reconstructions of our specimens. So it's kind of like focusing a spotlight on a very specific plane within that sample. While with widefield microscopy, you're just illuminating the entire stage.
So confocal microscopy gives us this clearer, more focused view, while widefield microscopy provides a broader perspective, but maybe less detail.
Exactly.
Okay, that makes sense. Are there any other kind of advanced microscopy techniques that are used for fluorescent imaging?
Oh, certainly. We also have techniques like FLIM, or fluorescence lifetime imaging microscopy.
Wow.
And that actually measures the time a fluorophore spends in that excited state before it emits light. That gives us a bunch more information about the molecular environment of the probe, and it can be used to study interactions between molecules or even changes in cellular conditions.
It seems like we've moved beyond simply visualizing these structures. We're actually probing the dynamic interactions in these molecular landscapes within cells, which is pretty awesome. What about multiphoton microscopy? I've heard that allows us to see deeper into tissues.
That's right. Multiphoton microscopy uses longer wavelength pulsed lasers to actually excite our fluorophores. And this allows for deeper penetration into tissues with less scattering.
Okay.
So it makes it really useful for imaging thicker samples like brain slices or even whole organs.
So multiphoton microscopy really opens up this window into the depths of biological tissues, revealing those structures and processes that would otherwise remain hidden from our view. It's incredible how these various microscopy techniques, all coupled with these fluorescent probes, have completely revolutionized our ability to explore the inner workings of cells and tissues.
It really is amazing.
It's like we've gone from peering through a hazy window to having this high-definition, multi-dimensional view of the cellular world.
Exactly.
But this journey of discovery, it isn't without its challenges, is it? We kind of touched on some of the limitations of these fluorescent probes earlier. Can you elaborate on some of the challenges that researchers actually face when they're working with these tools?
Sure. One big issue is photobleaching, where those fluorescent molecules gradually lose their ability to emit light when they're repeatedly excited. This can limit the time we have to actually image a sample before that signal fades away.
So it's like a race against time to capture the data before that fluorescent signal dims. Are there ways to minimize photobleaching then?
There are. Researchers can use various strategies like reducing the intensity or the duration of that excitation light,
Uh,
using anti-fade reagents, and optimizing those imaging conditions to try to minimize photobleaching.
so it sounds like a delicate balancing act.
It is.
Between capturing enough signal to get a clear image, but also preserving the integrity of those fluorescent probes, what other challenges come to mind?
Another hurdle is autofluorescence.
Okay.
And that's where some cellular components just naturally emit light, even without being labeled with a probe.
Ah.
And this background fluorescence can interfere with the signal from the specific probes we're using, so we have to carefully account for it.
So it's like trying to listen to a faint whisper in a noisy room.
Yeah.
The autofluorescence can drown out that specific signal that we're trying to detect. Are there any other pitfalls that researchers need to kind of be aware of when they're designing these fluorescent imaging experiments?
Well, one more challenge is fluorescence overlap.
Okay.
That happens when different probes used in a multicolor experiment, they emit light in similar wavelengths. And this can make it difficult to really distinguish between those different signals. So it requires really careful selection of probes with well-separated emission spectra.
It's like trying to decipher a message that's written with different colored inks, but they've all kind of bled together.
Right.
The overlapping signals can kind of blur the boundaries between different structures or molecules that we're trying to actually visualize.
Exactly. Careful experimental design and using the appropriate controls is really crucial for minimizing these challenges and making sure we're accurately interpreting the data from our fluorescent imaging.
It's clear that while fluorescent probes offer these incredible insights into the cellular world, they also require a really high level of expertise and just meticulous attention to detail to avoid those potential pitfalls.
That's true, but the rewards far outweigh the challenges. Fluorescent probes have truly revolutionized our understanding of cell biology, and the possibilities for future discoveries, they seem limitless.
I couldn't agree more. Yeah, it really is amazing to think how these tiny glowing molecules have really unveiled such a vast and intricate world, just all within our cells. So as we're wrapping up here, I'm really curious, what excites you the most about the future of fluorescent probes and cellular imaging? Like, what are some of the most promising avenues for future research and development?
Well, I think what excites me most is the potential to actually translate these powerful tools from basic research to clinical applications.
Oh, wow.
Imagine using fluorescent probes to diagnose diseases earlier, monitoring treatment responses in real time, or even delivering targeted therapies directly to diseased cells. That'd be incredible.
It really is incredible to think that we could actually harness the power of fluorescence to kind of guide these medical interventions with that level of precision.
Yeah.
Are there specific areas where you see this technology kind of making the biggest impact maybe in the near future?
Well, one really promising area is in cancer research. Fluorescent probes can be used to image tumors, track their growth, and assess the effectiveness of different therapies. So many different applications.
So we could potentially use these probes to actually personalize cancer treatments.
Yeah.
Like tailoring them to the very specific characteristics of each individual's tumor.
Exactly.
That's amazing.
Absolutely. And beyond cancer, there's also immense potential in using these probes to study and treat neurodegenerative diseases, things like Alzheimer's and Parkinson's. We could use them to visualize the accumulation of those toxic protein aggregates in the brain, monitor neuronal activity, or even deliver drugs directly to those affected brain regions.
Wow, it's like opening up this whole new frontier in neuroscience, allowing us to see and understand the brain in ways that were never before possible.
Precisely. And as we continue to develop more sensitive and specific probes, along with more advanced imaging technologies, the possibilities for understanding and treating these complex diseases are just going to keep expanding.
It's truly an exciting time to be working in this field. It's like witnessing the convergence of biology, chemistry, physics, and engineering all coming together to unlock the secrets of this cellular world, and then translating those discoveries into these tangible benefits for human health.
I couldn't agree more. It really is a privilege to be part of this ongoing journey of discovery and innovation.
Well, on that very inspiring note, I'd like to thank you for joining us on this exploration of fluorescent probes and their remarkable impact on cell biology research. Until next time, keep asking those questions, keep exploring, and keep marveling at the wonders of the microscopic world that lies within each and every one of us.